Hello, I'm Brett McGregor, and I'm here to tell you today about our lab's entry into the CMAP Drug Safety Challenge. My poster is titled Gene Expression Signature-Based Machine Learning Classifier of Drug-Induced Liver Injury. Some of the major concerns in drug development and discovery is the toxicity and safety of these drugs. Since the liver is the main location of drug metabolism, it is more vulnerable to drug toxicity. Hepatic dysfunction or abnormalities caused by drugs is referred to as drug-induced liver injury, or DILI. DILI is a primary liability in drug development because it is often not detected in the current preclinical models. This has led to a need to develop alternative methods of predicting DILI. There are many factors which can contribute to DILI. Some of these factors are related to the drug, such as reactive metabolites, or a more direct effect, such as engaging the immune system. However, many factors are also unique to each patient, making prediction of DILI a complex challenge. For this challenge, we focus on 4 different classifiers of DILI. DILI 1, which involves drugs that had a severity score of greater than 6, DILI 3, which involved drugs that were either withdrawn or had a box warning, and DILI 5 and 6, which involved more minor endpoints in DILI. Our drug list was split into 422 drugs for training and 195 drugs which we had to predict what their DILI scores would be. The information we have on these drugs to help form our predictions include chemical descriptors from MOLD2, host factor information from FARES, pathway perturbation from Tox21, and cell line expression data from Link's L1000. For this talk, we'll be focusing on the cell line expression data. The objective of our study was to develop traditional machine learning-based prediction models for our 4 DILI types. We did this using various combinations of predictor sets, testing them using support vector machine, generalized linear model, random forest, naive Bayes, neural networks, and recursive partitioning and regression trees. All of this was performed in R using Caret and other machine learning related libraries. The cell line expression data used in this study from L1000 links included 6 cell lines, the PHH or primary human hepatocytes, and HepG2 or liver cancer cells were a major focus as they are directly related to the drug-induced liver injury. We also used HA1E, the human kidney cell line, A375, a skin cancer cell line, MCF7 as a breast cancer cell line, and PC3 as a prostate cancer cell line. Not all drugs were tested across all 6 cell lines. However, 247 drugs were shared across all cells. This is the majority of the PHH tested drugs. 164 drugs were not tested in either PHH or the HepG2 cell line. Another thing of note is that the PC3 and MCF7 tested drugs are the same, which allows for a gender comparison. We analyzed the 12,000 genes for predictors in a variety of ways. For each cell line, we administered a z-score cutoff of 1.96, or 2 standard deviations, and performed a t-test between DILI positive and DILI negative drugs. Using a p-value of 0.01, we identified which genes could be considered predictors of DILI to train our models. We also used Kruber merging to combine the expression profiles of a drug across all cell lines it was tested in. The top and bottom 100, 250, 500, and 1000 ranked genes after merging were selected as a drug's transcription signature. Using a Fisher's exact test with a p-value cutoff of 0.01, these transcription signatures were tested between DILI positive or DILI negative drugs, and we were able to filter out non-significant genes. We also tested an integrated model using our t-test genes from PHH and HepG2 combined with FARES and Tox21. The Kruber merging method used to produce our drug transcription signatures across the 6 cell lines uses ranked genes to reduce the distortion. For each cell profile a drug has, the 2 closest ranks will be merged until each of the 12,328 genes are ranked. Using this method, we reduced 2,844 drug profiles in 6 cell lines to 611 drug profiles, and selected the top and bottom 100, 250, 500, or 1000 ranked genes as a drug signature. Here you can see the training performance for our models predicting DILI-1. Multiple model types were tested for each selection of features, but only the best model type is shown here. Overall, our merged top and bottom 1000, along with the PHH tested genes, performed the best at predicting DILI-1. In the training performance for DILI 3, our Merge 1000 model performed the best again. However, all transcription features showed some ability to predict DILI 3. This may indicate that changes in transcription are effective at predicting drug warnings. In predicting DILI 5, our merge models outperformed our single-cell models. The Merge 1000 model performed the best in this case as well. We see a similar pattern in DILI 6, where the merge models outperform the single-cell models. However, this time the Merge 500 model performs the best. Shifting towards our integrated model, since females have been reported to be at a higher risk for DILI, we want to know if integrating gender-specific features could improve our models. Our integrated model combines FARES reporting ratio and Tox21 with gene expression from PHH and HepG2. The male data includes the PC3 cell line and male FARES reporting ratio, and our female data includes MCF7 and female FARES reporting ratio. These are still a preliminary look at these models, but right now it looks like it decreases our model performance, which needs to further be examined. In summary, gene merging improves our models compared to cell-specific modeling. Typically, the top and bottom 1000 gene model performs the best. Our integrated approach is still a work in progress. While this poster only addresses the transcription data, our work with the other types of data will be presented in a short oral presentation by Dr. Herr. Our future works include exploring voting or other integration methods to incorporate these other data types with the transcription data, better understanding the data we are dealing with, such as the identity of predictors, to possibly identify an underlying mechanism of DILI, and also to understand the role that gender plays in drug-induced liver injury. With that, I would like to thank my lab members and thank you for your time.